Hi everybody. I hope everybody's okay. I just opened and it closed. Today, see, it was open already. Why it didn't show? Today, I would like to talk about yeast to hybrid system. This is going to be an interesting point. I'm gonna close this one. Make it full screen. Yes, uh, it's an important topic. A lot of people are working with proteins, and understanding proteins is not easy. So this is one of the methods that scientists use to understand proteins. So this was a homework actually for one of our classes, microbial experimentation or something like that. So microbiology techniques or something like that. It was a class. So just let me start. Experimental microbiology. Now I remember, experimental microbiology. So this was my report actually, so I'm actually make this a little bit visible. Okay, now I guess this much better. So it's actually a very interesting topic. So the very high tech experiment. The idea is to understand proteins, and to do so, we use organism yeast. You cannot do this in bacteria because yeast is an eukaryotic organism, and it has many advantages over bacteria, and I will explain this and why proteins, why try to understand proteins. But I mentioned earlier, bacteria has a different mode of regulation, and our cells are more like yeast cells, and yeast cells have lots of proteins, and to understand what these proteins do is not easy. I mean, the cell is full of proteins, and their structure is complex, and the worst part is they don't do much by themselves. I mean, oftentimes you see 2 or 3 or 4 proteins come together and carry out a function. So you never find a single protein, doing much in the cell. So there is always this cooperation, this protein complexes just coming together. That just the different say this is protein A, protein B, just some sort of connection there, kind of getting locked with each other, and make some sort of the complex or a system or a machinery, I should say. It's not easy to understand, how these proteins work. So because I have to do this introduction, the rest is easy. I mean, in terms of the techniques and the methods, and I will explain them. But the idea, I mean, the idea is to understand proteins. Same thing with drugs and or some of the food that we eat, proteins like vitamins and other things. Oftentimes they work on proteins, you know, whether they are on the cell wall or cell surface or inside the cell, one way or other, you know. Most of the time, what we eat and what we do changes protein configuration, changes protein concentration, changes what these proteins do in the cell. This is called protein signaling, enzyme activity. There are various names, but just like I said, first thing first, we have to understand these proteins. They are there for a reason, and they do many different jobs. And this one is particularly about turning genes on. How to turn a gene on. So, with this introduction, this is yeast two hybrid system is a method developed to understand the relation between two hypothetical proteins, whether they bind to each other or they come together for a specific function. You don't know, but you just want to test. You just want to see if you can understand whether these two proteins come together and carry out their function. Like I said, in the cell, many of the proteins work in these complex structures. Remember the just some like the movies. Remember the Autobots and Transformers. Like many of them come together and and become a bigger robot or something. This is like that. And why proteins are important? Because they do many important jobs. I mean, some of the proteins are for food we consume. When we say proteins, we oftentimes think of them as food, but eventually what's inside proteins are amino acids, and once we digest these proteins, we get those amino acids. Some of them we cannot produce. We need them, and then we generate our own proteins. So proteins are in the plants. Proteins are in the fungi, in the yeast. Anything alive has proteins. It's just even viruses. We know that certain amount of proteins they have on the envelope, or on certain parts that they produce inside a host organism or a host cell, like envelope proteins and such. So let's start with the experiment. So let me just read just fast. This is my writing, my report. Baker's yeast genome, which is eukaryotic, includes many conserved genes related to basic cell functions such as respiration, response to environment, and utilization of nutrients, etc. in cells. Its ability to carry and maintain plasmids can also be found in many bacteria. Just So far, very easy. Plasmids, they're just pieces of DNA. Plasmids are often defined as short circular DNA fragments, usually less than 15,000 base pair long. These DNA fragments can be designed in such a way to carry genes of interest with help of endonucleases, which are enzymes that recognize and cut DNA at certain sites, and often combination of 4 or 6 base pairs. So the plasmids are already in nature. I mean, humans have not invented them. They are in bacteria, and then if you remove them from bacteria and cut and insert some other genes in them, you know, they are still plasmids, and you can carry, some functions from bacteria A to yeast, or from yeast to bacteria. So it's like, remember, it's like, for computers you have floppy disks and memory keys, it's a short piece of DNA, and you can cut DNA with certain enzymes, and then, paste and cut, paste and close, and so there are enzymes to do that, and we call them endonuclease. They just cut DNA at certain locations, certain letters, etc. Letter combinations, base combinations. Specific endonuclease cut DNA fragments with the same compatible ends, and then can be ligated back, like merged back to each other, resulting with a joint fragment consisted of fused sequences which may encode a protein or regulate a gene. So you can cut these DNA fragments and place them in a plasmid, and then you can do some experiments with it, just basically. Plasmids carrying such fragments are often used to express foreign proteins or restore native genes in mutant genotypes in both yeast and bacterial cells. It's like installing a program. I mean, it's just basically, it is what it is. Like installing a program in your phone, app. It's the same thing. Plasmid is, like a app that you just can put in a cell. It's just, now everybody is like typing apps and making new apps, right? App for this and app for that, and imagine nature already invented it, with this thing called plasmids. The bacteria, were doing it for maybe billions of years, billions of years. So it is important to learn biology, because biology is the highest form of technology. Highest form of technology. Biology is the highest form of technology, because even now, the way cells work, many computers can't still. And that's why we are trying to understand, because it's these proteins oftentimes act like a processor, because they are built with many components. Component A, component B, component C, right? So we're trying to understand, how these proteins come together and make a composite system. So eukaryotic protein genes, not bacteria, are structured in a way. Eukaryotic protein genes are structured in a way that they are controlled by upstream sequences of the open reading frame that encodes a specific protein. These sites are called cis-acting elements, and they control the transcription of the downstream DNA gene sequence upon receiving stimuli or molecular signal. So it's like a switch box. There is this gene in eukaryotic cells. There is the gene A, and then there is the switch box, the switch system, the box of the gene. and sometimes you can change the box, the gene. Okay, the box turns on the gene, but you can sometimes if, if you know the mechanism, you can change the box, and so you can decide how the gene is activated and such. So these sites are called cis-acting elements, and they control the transcription of the downstream DNA gene sequence. Transcription meaning activation, just turning into RNA. Upon receiving stimuli or molecular signal, of course, when the time is right, then the DNA gets the signal and turns the gene on, and there are some genes that are used once in a lifetime. So when the time is right, the gene gets turned on. Some genes work all the time. Some genes just we have tumor suppressor genes. We have genes that work when we are a little tiny cell, as an embryo, for example, the genes. For example, our hands are not like this. Now we have bones, but there is a gene just that makes these shapes. It's like birds. Sometimes there are mutations and people have connected fingers, connected toes. And that's a mutation. You understood, So genes work. And there's a mechanism that makes them work. So very simple. So just by watching the time, it's important because this is once the time is right, stimulate, the signal. This is often carried by kinases or enzymes in a signaling cascade, where final signal protein starts activation of transcription via recruitment of RNA polymerase, which synthesize mRNA for translation. When you want to turn the gene on and synthesize mRNA, you need RNA polymerase, because you need to synthesize mRNA, and this is the enzyme to do so. And to get this enzyme into work, you have to recruit it, to the DNA. You have to bind RNA polymerase to the DNA, so that it synthesizes mRNA, which is a copy of the gene, to be translated into a protein. Okay, so it's like this is the RNA polymerase is like the copy machine. It's like the scanner. This is DNA, RNA polymerase. It scans and makes a copy, and that copy is usually called mRNA for translation. Upon reception of environmental cellular cue, like when the time is right, for example, think of a seed, in the ground, it is dark. Think of a seed, in the ground, it is dark. It does need what? First, roots, For roots, you don't need to see the light, The seed needs to generate, develop roots. For this, you don't need light. But once the roots are established, then you wanna produce leaves and the rest of the plant. The stem and leaves and the chlorophyll molecules and photosynthetic proteins. And for that, you need signal, light. When the seed senses the light, it starts, production of these photosynthetic proteins. This, so example. So upon reception of environmental and cellular cue, relay of signal is usually carried out by phosphorylation of downstream cascade proteins by adding phosphate group to their conformation, changing their shape so that activated protein can either phosphorylate another protein or bind to gene-specific regulatory DNA site to initiate expression of required proteins. So this signaling mechanism, oftentimes it is not understood well. Signaling can either be done by protein-protein interaction, or some proteins can go directly into the nucleus and attach to the DNA or the DNA activation systems to initiate expression of required necessary proteins. Again, it's all about proteins, like whether enzymes or a product, because the same, you need muscle, protein, or just they do many jobs. Enzymes, digestion. So during signal transduction, during this cascade of signal system, during signal transduction, proteins can also bind and get linked to each other, forming complexes in order to carry signal further to a target protein or DNA regulatory site binding protein. It's all about getting the signal to its target. There is light, and the cell needs to synthesize certain proteins. There is the signal. The cell needs to generate digestive enzymes. In this case, it is about digestive enzymes. Because, for example, you have food in your stomach. What does it mean? And you have to digest it. You have to synthesize these digestive proteins. Example. Whether it is digestion or something else. For animals, it is usually digestion. Such complexes can also start transcription by recruiting RNA polymerase II upon binding to a regulatory site. So it's about recruiting RNA polymerase. Again, this is mechanism, to get the protein synthesized, whether it's an enzyme or something else. You need a combination of proteins, to get the job done. Either signaling proteins or often often proteins that bind come together and make complex. This is called the binding, affinity testing. That just in chemistry it's called covalent bonding. It's very strong in chemistry. Covalent bonds. Like the structure of DNA backbone. The DNA backbone is supported by covalent bonds. The phosphate bonds. But for example, DNA is two strand in between. Those bonds are rather weak, so you can separate the two strands of the DNA. And same thing. These proteins, when they are in right conformation or configuration, they just bind to each other, not like pinned and glued irreversibly, which some toxins do. That's why people suffer from poisons oftentimes these are irreversible, reversible. They bind, and then after a time, they release back into the solution. They separate. So recruiting unit is called DNA binding protein, and it could be composed of different functional domains. So in this case, there is a complex. There is protein A, there is protein B, and once A, B come together, it recruits the RNA polymerase to make the copy of the gene. So two domains, they call it, activation domain and binding domain. The one protein binds to the DNA, and the other one activates the RNA polymerase. And so the four of them come together: DNA, activated protein, binding protein, RNA polymerase, and then the protein gets produced or synthesized. And you can construct this system, this activator protein, binding protein, by utilizing these two proteins, to turn on a gene, so it's about this. That's why it's called yeast two hybrid system. So you generate one hybrid by using activator protein and another hybrid with the binding protein. So if those two proteins of interest are forming a unit together in the cell, they will bring the activator and binding proteins together, so you will have a expression of a gene. But say you are studying X gene and Y gene, and they work together. You are thinking, you don't know if the X and the Y proteins are working together in the cell. They bind and they form a complex, and you can test it with this system. You bind X protein to the activator, Y protein to the binder. That's why it's called hybrid. You hybridize them, you fuse them. You fuse the two together, and if they come together naturally in the cell, you bring the activator and binding protein together, thus expressing a protein or an enzyme which you can see by color, etc. So this is one of those experiments, and then it's done with the yeast, and in several ways. I will explain them. So it's kind of I hope everybody you can. There are some videos on internet. I mean, this is not like top secret. There are some videos on internet explaining yeast to hybrid, but this is my way of explaining it. And then at the time I think my professor didn't like very much how I explained it at the time, or maybe he was expecting a much better explanation from me. I don't know, but this was my best, and I did my best to explain it in the year 2007. Standard methods such as co-immunoprecipitation are used to analyze protein-protein interactions with help of insoluble antibodies that can be coupled to a subject protein. There are some other methods. Precipitated complex then can be analyzed via Western blotting method, etc. So you can study same protein-protein interactions with antibodies. You can tag them, and then try to get a reaction or something. What is the downside? It's a biochemical process and cannot be performed in live cells. See, doing an experiment in vivo is different. Doing it in vitro is easy. Doing in vivo, like where the action is happening in the cell. In doing an experiment in live cells is not easy. So this is a live cell experiment. I mean, it is not a chemical one. It is a it's a biological experiment. You are literally trying to see if the proteins meet inside the cell, and then, and if they are together, they just generate the enzyme that gives you a color. So what are the disadvantages of the standard methods like these chemical methods? There's biochemical techniques like poor antibody fidelity. The tags sometimes they don't work properly, and protein contamination it just affects the reliability of the real complex. So in yeast to hybrid systems. So this is a model. This is, like I said, a lot of people don't know this. This is called yeast to hybrid system, and it works. So studying yeast is a great privilege, and if you are working with proteins, you need to know these methods. If you ask me. And now we need all different kinds of proteins for food, for medicine, for other research areas. Whether you are interested in astrobiology or something else, to understand proteins, maybe you wanna listen to this and learn. In live cells. So in yeast to hybrid systems, interactions can be studied in live cells in a relatively short amount of time with less biochemical processing and errors. So you see, less biochemical processing. That's important because, like, for example, doing a Western blot experiment takes 1 week. It's just like all the washing and with antibodies and nitrocellulose and transfer and SD pages and everything. In this method, there are two constructed hybrid proteins. When in the right conditions, two form a complex and interact with RNA polymerase 2, to recruit it to the target regulatory site to initiate expression of a reporter gene, which gives you blue color. Reporter gene means like changes color, and then you understand. Okay, it turned on. Whose activity can be measured via biochemical means, usually changing colors, etc. Complex forming protein-protein interactions can be tested with this method. So again, this is just basically a method to understand protein interactions. Very important, because our life is full of proteins. Whether they are a source of food or source of medicine, and all the drugs too. All the drugs target proteins, because they have such a complex, in nature. Some drugs like bind to this part and some to other part. So you can never guess. This is just yeast. So this is let's continue. Yeast strains can be engineered to have specific reporter genes and regulatory binding sites. Reporting genes means like if you turn the gene on, it just generates a enzyme or changes color. It's called reporter gene. It's not as significant like metabolic activity. It's just a gene. It just tells you that it turned on. And regulatory binding sites. So if you have a reporter gene and you can play with it, you can change the like a switch box. You can change the how to turn the gene on and off, and etc. So this can be accomplished with the help of specifically constructed integrating plasmids that are integrated in the chromosomes in order to replace a gene or restore a defective copy by a recombination event that normally occurs during yeast cell division. So there are kinds of plasmids. Yes, there are the regular plasmids, and there are plasmids that are integrated into the genome. These are called integrating plasmids. That's how you generate mutants. And if you construct a plasmid with a reporter gene on, with a reporter gene and a special regulatory binding site, if you construct this plasmid and insert it into the yeast, then you can insert other plasmids and generate some proteins, some DNA activating proteins, and see, do experiments with that. So excuse me. So so it's called transformation. There are ways to do it. So that is understood, I guess. So I said, the two types of plasmid. One, you can place it inside the cell, and then there can be so that plasmid normally washes away if it is not necessary. Normally cells don't keep plasmids because it's a burden for them. But if it is integrated plasmid, it stays in the cell. So that's why these plasmids can go in and out of cells. So let's understand that first. So there are two types of plasmids. Just one you can have the gene on, and just whatever you have constructed, you can insert into a bacteria, into a yeast, and express the gene, and then after a certain time, it just even the cell get rid of it if it is not necessary. So if you can get these plasmids express these hybrid proteins, you can do experiments. Under right conditions, hybrid proteins will interact by forming a complex, therefore allowing RNA polymerase to the reporter gene, and which gives you a color, or some sign. So this is actually very complex, but it gets more complex after this. But I'll try to summarize it as much as possible. Even I have read it before, like 1 hour, 2 hours earlier, how to summarize, make it digestible. So hybrid proteins are designed in a way that DNA binding protein complex does not start transcription on its own. They're called the two pieces. The one is called bait, the other is called prey. DNA binding domain bait should be dependent to second hybrid protein, to which includes a transcription activation domain or protein of interest. So you have to construct hybrids in a way that that they don't come together without your proteins. And you're trying to see if they come if your proteins make a complex only when they are together. So there's one thing to consider: prey and bait. That's one is called bait, one is called prey. Scientists often fuse genes of interest with special DNA binding proteins in a way that it can be tested for complex forming ability with another subject protein, such as those expressed from a cDNA library. These genes are present in DNA libraries, and you pick and try to see if these two proteins interact with each other. this way subjects I did a typo there. This way subject proteins will form functional complex if they interact and bind to each other, confirming that they can be the members of the same signaling pathway. For example, say there is this signal Q, environmental signal, light, temperature, whatever, and the protein A it gets affected, and then protein A goes and binds with protein B, and then make a complex, and then and continues the function, or continues the signal, relays the signal. Just that's why it's called yeast 2 hybrid. You're just trying to understand the relation between proteins. In this experiment, we will compare different proteins and plasmid constructs in P-Lexay yeast to hybrid systems. So that's the name of the gene, Lexay. In this system, in this system, bacterial lactose utilization gene LACZ is integrated into a yeast strain with an artificial Lexay promoter site regulating it. So like I said, normally they took this gene normally yeast doesn't have anything with the lactose, but they got it from bacteria, and put it on a DNA and then integrated it into a yeast. It's just like I mentioned earlier, there are types of plasmids, and this is an integrated plasmid. They replace the gene with this gene, LACZ gene, and this gene has an artificial promoter site. This is the switch box. This gene, they got it from a bacteria, and it has an artificial switch box promoter site. Okay, Lexay. It's a special box, and then so if you can turn, if you can prepare the system to turn this site on, then the gene will be turned on. It's just because this is important. This is the most important part. This is the gene. You want to turn this on, okay? Which will give us the blue color. But this is the box. This is the switch box, and then we're gonna learn how to work it, how to run it. Lexay promoter site regulating it upon interaction of because normally you cannot do this much in bacteria. That's why it's like a hybrid system. You get the gene from bacteria and put it in the yeast, and then try to understand how eukaryotic cells work. So they changed the original promoter of this gene, and then put a proeukaryotic promoter, Lexay promoter site, regulating it. Upon interaction of a hybrid protein complex with DNA, RNA polymerase DNA and RNA polymerase 2, Lexay expresses beta-galactosidase enzyme, which is a digestive enzyme. That can react with the substrate, something that gives color, such as X-gal, this material, chemical, this substance. The reaction results in the liberation of X substance, which turns colonies into blue color. Once you turn the gene on, it produces this enzyme, and then the enzyme breaks down this substrate called X-gal, and then when you break down X-gal, you get X, I think, which is blue color. It turns the colony into blue, just basic. It's like a basic digestion, Other substrates such as OMPJ are also used where products turn cell suspension into yellow color, which can be quantified via spectrophotometer to measure promoter activity. There's some other color changing options, but this is xgal substrate, beta galactose enzyme. So it's just so materials and methods like this is mostly now let's understand the plasmid. I'm just gonna briefly explain it. You get the plasmids from bacteria, E. coli, Escherichia coli, DH5 alpha, alkaline lysis method, overnight grown cultures, and then the plasmids have ampicillin resistance genes in it, so LB media with ampicillin. So you select the plasmids from E. coli, and then this is the gene. This is the plasmid that is carrying this one protein, one of the proteins, Lex-A protein. Remember, this protein is necessary to bind to the switch box in the yeast chromosome, in the yeast DNA. Now we are talking about plasmid. Now we're talking about genes and proteins. So the plasmid is carrying this gene, and it is necessary. Lex-A protein and gene. It's on a 2-micron shuttle plasmid carrying ampicillin resistance gene, along with the His3 gene. And ampicillin you need to select for bacteria, and His for yeast, because you're using the same plasmid, and if you're using it in bacteria, and I think beta-lactamase, I think is the name of the gene, ampicillin resistant gene, beta-lactamase. I think if the plasmid has beta-lactamase, then it breaks down ampicillin. It survives. Otherwise, other cells can't. That's why. So they put the beta-lactamase gene inside the plasmid, so that they can work with bacteria or make more plasmid. And another selector, His3 gene. So you can select in bacteria and yeast. So it's a low copy plasmid. It has what else? It has alcohol dehydrogenase promoter following Lex-A DNA binding protein gene. Now I'm gonna show just a brief figure. First, we have the ADH promoter. See, this is another switch box in yeast. This is a good promoter that works well. ADH promoter, alcohol dehydrogenase, and following this promoter, remember, it was a switch box. It is the switch box of the gene, and Lex-A is the gene. This is one gene that in our experiment that is necessary. So this is one plasmid, and then I'll show other plasmids too. There are like a couple plasmids, and this is just Lex-A plasmid, and a multi-cloning site. So you can insert the protein of interest next to this DNA binding protein gene. So you're gonna make a complex. Remember, just DNA, protein A, protein X, protein B, and protein Y. And eventually RNA polymerase. So we're trying to bring 6 things together with this experiment. To be honest, like 4 proteins, in 2 hybrids, 4 proteins. That's why it's called Yeast2 hybrid. One hybrid, second hybrid, and then these hybrids if they are right, if they work together, they bind to DNA and and recruit RNA polymerase. So total of like 5 enzymes. Imagine, like in the cell, like 5 enzymes on top of the gene on DNA. That's why it is amazing. I mean, it's just like 5 proteins. Like I'm just, giving you the numbers here. I mean, just that's how the experiment. And then ADH terminator sequence. So if you want to generate, it's if you want to generate a plasmid, yeah, maybe you can use this method. Yeast alcohol dehydrogenase, let's say DNA binding protein gene, multi-cloning site, and ADH terminator sequence. BMH, I think it was BMH1, was used to construct, let's say, SNF1. So that is one hybrid, let's say SNF1. Second one is, let's say, SERB11. Those are that's the second plasmid, that's the second construct. SNF1G was used as a negative control that doesn't activate transcription when fused and co-expressed with the lexase protein. So this hybrid doesn't do anything. I mean, when you combine lexase and SNF1 protein, which is a yeast protein, in this experiment we are trying to understand the relation between SNF1 and SNF4. So in the plasmid you will see there will be another construct with SNF4. But this combination alone doesn't do anything. So that's why it's a control, negative control. So it doesn't activate transcription, But this does, this is positive control. Lexase and SERB11 fusion, it does recruit RNA polymerase. It turns the gene on. This is a control. Lexase, SERB11. That this fusion acts like normal. SNF1 gene was used as a negative control that doesn't activate transcription when fused and co-expressed with the lexase protein. Let me just briefly, let me see how many minutes. 52 minutes. Oh my God! Maybe already. Actually, I'm already just finished. So in a second plasmid carrying the other protein, plasmid activating domain, another plasmid carrying the protein of the activation domain, along with LU2 yeast selection marker, because you cannot insert the plasmid without the marker. Otherwise, it won't keep the plasmid. It'll wash out. To be able to keep the plasmid, the plasmid should make the cell prototroph. Otherwise, it's no good. With the LU2 gene on the plasmid, you can grow it you can grow the yeast on a media that lacks leucine, and then it generates leucine and becomes prototroph. It's called yeast selection marker. So this is another Piget SNF4. So this is another construct. So lexase SNF1, So these are the constructs: lexase SNF1, lexase SERB11. These are all different plasmids. Lexase SNF1 plasmid 1. That's another plasmid. Lexase SERB11, and then this is the structure. See they just the multi-cloning site. It is inserted there. After the lexase part, see the multi-cloning site. That's what it does. You clone the gene here. That's why it's called multi-cloning site. So this is second construct, third construct, and then this is the fourth one. The Piget SERB11. constructed, fusions were constructed, so the hybrids. C-level protein acts as a transcription activator on its own when fused with DNA binding protein. So it's kind of a control, positive control. You don't need a protein interaction to turn the gene on. It just activates on itself with the DNA binding protein. So, plasmids and inserts were confirmed with BMH digest. See, plasmids and inserts, I mean the genes, see from the DNA library, were confirmed with BMH1 digest. If you digest with the BMH1 enzyme, you should see the enzyme, the gene. I'm sorry. I mean, you have the plasmid. You digest it with the BMH, and then if it cuts, and then you should get the gene that you put it in there. And you can understand it with various methods and by the size and everything. All constructs are translational fusions, allowing continuous reading frame ending at terminating sequence of plasmid. So reading frame, remember, so the plasmid has a starting point and ending point, and then if you insert the gene correctly, in the frame of the plasmid, it should work. Translational fusions, which is, like, remember, ATG is like methionine. Every protein transcription, that starts with ATG, methionine. We all know that, protein. That's the first thing the ribosome, reads, oftentimes, 99% of the time, the proteins start with methionine, which contains sulfur. So I'm just gonna read it. Yeah, this is the kind of the structure of the. You should see the backbone. This is the general plasmid, PBR 322. That's the ampicillin, beta lactamase gene here, resistant ampicillin resistance. Histidine that is inserted for yeast selection. It codes for the histidine amino acid. To make the cells prototroph. And this is the lexay. This is the hybrid system. This is it's a protein A, and then you bind. This is one of the. Remember, this lexay protein is necessary to turn your reporter gene on. You need this is part one of the two proteins, and then you insert your protein of interest here, into the multicloning site, with this restriction enzyme, BMH site, endonuclease enzyme, and then you can insert your gene of interest here and generate your hybrid protein. Protein A, protein B fused together. Just fused together. And this is the total 10,000 base pairs. It's a small plasmid. It's just basically. So it's getting over an hour, but I'm just gonna make this fast. I think it is understood. So there's a plasmid one, some 2 micron, carrying histidine. Second plasmid carrying the second activating domain. This is second plasmid, GAL4 activated domain. Third plasmid, plexay and SNF1 protein of interest, and the fourth plasmid, activated domain. So this is binding protein, DNA binding protein. This is activating domain. So these two should come together, to turn the reporter gene on. If these two come together, we're trying to understand, if SNF1 and SNF4 for some reason work together in the cell. We don't know. That's the question. We're trying to test if these two proteins, SNF1 protein and SNF4 protein, we got these from DNA library. We're trying to understand, the effect of these genes, SNF4 and SNF1. What do they do? Do they come together? Does SNF1, make a complex with SNF4? Do they come together? We don't know. So, and if they come together, they bring these two proteins together too, which gives you blue color. End of story. Very simple. If SNF1 and SNF4 they come together in the cell for a specific function, hypothetically, we don't know. If they do, then since they are fused with, let's say, SNF1 is fused with, let's say, and SNF4 is fused with another activating protein, these two will run the reporter gene, and you get a color, and then you will say that, okay, SNF1 and SNF4 works together. They got so close so that these two came close. So these two came close, and activation worked, and then you got the reporter gene turned on. You got the enzyme, and then and then the substrate was broken down, and etc. And this is the control, and this automatically on its own works the reporter gene. Positive control. See, in the experiments you have to have negative control, and a positive control, regardless of the experiment. You have to in all life, what should it look like if everything goes right, and what should it look like, if one is missing or other, So it's called negative control and positive control. In negative control, you expect negative results. Very easy. In negative control, you expect negative results. In positive control, you expect positive results. Just very like you're baking at home. What do you expect? In the negative control, you're baking a cake. You don't put the leavening or egg or the carbonate, soda, and that's negative control. If you don't put those, the cake won't rise, basically. And you're trying to, for example, test whether 3 eggs, plus the soda makes a better cake. Then you have to have a positive control. You have to have regular cake, with 1 egg and 1 baking soda, and then you can test, 2 or 3 eggs or 2-3 bags of baking soda. So that, see, oftentimes it is very important in science. Positive control, what something should look like normally, so that's why it is hard to understand our lives. We don't have a positive control. We don't know what our normal is. This is normal. If actually, in other terms, positive control means normal. So trying to understand what is my normal, what is your normal. So that's important. In science, in experiments, you have to have a normal, and you have to have a negative. You have to have a normal. Positive control. Very simple. You have to know your normal in life, whether just you are a child, you are a parent, you are a spouse, what is normal, you have to know. So yeast strains, I'm just gonna so it's a special yeast strain with a special integrating plasmid. I'm just gonna go quickly. so these are haploids. So mating type alpha, mating type A, alpha. So they are similar, but mating type A, and this is alpha. In yeast, we have two mating types, A and alpha, and then and one has the one has the lex A operon region in the DNA. It has again the cell is also uretery positive. So that's a gene regarding prototrophy. See, it's mutant for adenine to histidine 3, Lu, tryptophan. That's how you make combinations. You know, this is just yeast genetics. It is URA3, and then has a LEX-A gene, LEX-A promoter, and a LEX-Z gene. LEX-A promoter, LEX-Z gene, and then these are mutations, extra mutations, GAL4 mutation, GAL80 mutation. So that some other element doesn't turn the gene on. They deleted these genes so it doesn't mess with this system. So you arrange these mutations so that they complement each other. Like this one has his mutation, this one has adenine mutation, for example. When you combine the two, if you can make prototrophic cells, and add or subtract what is necessary in terms of media. And then transformations, see the plasmid transformations. You insert the plasmids into the yeast. It's a lithium acetate standard transformation protocol. I did that many times. Synthetic complete media like with his and Lu selection. So I think the media doesn't have the complete media, since the plasmid has his histidine gene. The complete media, the synthetic complete media doesn't have histidine. The cell can the plasmid can produce it. So that's why it's called STD, SCD his, or SCD Lu. It means it's missing, minus his, minus Lu. It is missing those elements because, like I said in the beginning, the amino acids, some of them we can make. Our cells can make some of the amino acids. We have the metabolism. Some we cannot. Some we can make. If we have the gene, if we don't have the gene, we cannot. So you can arrange the yeast cells sometimes so that they can make their own amino acids and such. So you can work with that. And then we did a second transformation. That's important, like first plasmid and a second plasmid. So to co-express fused proteins. And then transformed the cells were assayed for beta-galactic acid activity via nitrocellulose filter lift method. I think we just plotted the nitrocellulose with X-gal material. So the cells can change colors. So this is the combination. See, the haploid transformation with the plasmid side, the typo there with plasmid side. So this is one transformation. See, this is synthetic complete media lacking histidine. It's a plasmid with the LEX-A protein, plasmid with the first hybrid, plasmid carrying LEX-A SNF1 hybrid, the number 5 plasmid carrying LEX-A and SERB11 hybrid. There's control, like negative control. And the second strain, and these are compatible. That you can mate these two strains, which is another way of doing this yeast to hybrid. You can do it by plasmid transformation, double transformation, or by mating of the haploids. So it's important, if you're working with haploid cells, you can also use this system. So this is the plasmid number 2, synthesizing activation the domain protein, and then plasmid number 4, synthesizing activation domain protein along with SNF4. So this is the second hybrid, and then in this experiment we expect we expect the combination of the two to work, because that's our working combination. You need activator, the binder gene, the activator, the binder protein, activator protein, and if SNF1 and SNF4 works together, binds together, makes a complex, then these activators and binders should turn the gene on and give us a color. And this is again control, and this is positive control. So this should to turn the gene on its own. So it's a working combination. This HERB11 acts like a activating domain. We said that earlier. It's this protein. So you have to be careful when you're choosing the proteins. If you're choosing an activator protein, it won't work. So you have to make sure it doesn't activate on its own. So you have to make sure you have to be careful when choosing proteins in this system. So this is a filter lift. This is haploid transformation with plasmids. Again, these are cells transformed with plasmids. See again the combination of the cell, the genetic. So that's the gene, LEX-A gene, along with LEX-Z. So this is the diploid. So the combination of so this is CTY10 and 5D. This is the diploid. So when you cross two haploids, you get a diploid, and then yes, no, I am sorry. This is not a diploid. No. Just next slide. Maybe I mixed the slides. Not diploid. I saw this one. This is not diploid. Mate A. See, it's my mistake. So that's why it's getting more than an hour. So co-expression of fusion proteins in the same strain were achieved by also crossing two transformed haploid strains. So if you put two plasmids into different haploids and then make a diploid, and then in the next figure now we have the diploid. These are the diploids. So when you cross them, the Y2 is a haploid, CTY is another haploid. When you cross them, and then depending on the proteins they have, you should have the working combination. Remember the combination. That just the 5 proteins should come together. The DNA proteins and in your proteins of interest, if they work together, if the 5 of them works together, then you get see the color change. Media is synthetic complete media were used to select for plasmids. See, another media was used to select for diploids, because once you made the haploids, and then now you have a different combination, prototrophy changes, and then this was another complementary media, synthetic complete media, missing histidine and leucine. This is more challenging, it's, see, this one's only missing histidine, this one's only missing leucine. And in the diploids, you have to be missing both of them, uh, so to understand, to select the diploid. Because only, if it is a diploid, then the cells will grow on a media missing both of them, If they mated, if the haploids mated, then they will become prototroph, and then so they won't need histidine or leucine in the media. In this one, this one has leucine gene, this one has histidine gene. Results, and so a single transformation of CTYY with plex A SERP11 resulted in blue color. Remember, this was positive control after filter lift assay. On the other hand, plex A control along with plex A SNF1 did not activate because they were themselves. It just didn't work by themselves. And with SNF1, SNF1 didn't turn the reporter gene on, even though it was connected to the binding protein. So these are negative ones, and then what was positive? CTY5. So this is the color you get. It's like a bluish color. Results of second transformation showed that plasmids were able to transform haploids into his-Lu prototrophy. So that's called prototrophy. When once the haploids mate, they change the prototrophy. So it's important, when working with yeast, when they mate, you have to make another synthetic complete media to prepare for the required prototrophy. So they complete each other. Again, results of second transformation. One first plasmid, again showing the plasmids again. Second transformation, we did second transformation, like you have one plasmid and then you put another plasmid, it's like it makes very challenging. So the first plasmid was selected with his, a second plasmid was selected with Lu, so when you put the transformed, the second, the cell with the two plasmids, you have to put them in, again, same thing, in a synthetic complete media missing histidine and leucine, because now you have first plasmid synthesizing his3 and second plasmid synthesizing Lu2, and to be able to understand that they are actually in the cell, then you have to have this plate missing histidine and leucine. So that is just to keep the plasmid, and that has nothing to do with the gene and operation. So this is just so the cells keep the plasmid. And let's see, in this plate we have this combination, 3 and 4, plasmid 3 and plasmid 4. So this combination should have the color, because it has the plasmid number 3 and it has plasmid number 4. If the genes are interacting, SNF1 and SNF4, if they are interacting, then you should have a color. Second transformation enable co-expression of hybrid proteins and activating transcription via interaction of CNF1 and CNF4 proteins. And, that's what we were testing. If these two proteins interact, what happens? The interaction means getting closer, so that those DNA proteins can get closer and combine and make a functional unit. So CNF1 CNF4 complex brought Lexay protein and GAD activator domain together at Lexay binding site, where Lexay gene was activated to express beta-galactosidase, figure 6. So just again briefly, CNF1 CNF4 complex, these two proteins work together in the cell. They came together. CNF1 CNF4 complex brought Lexay protein and GAD activators together to the Lexay gene operation site, and then the gene was activated and expressed beta-galactosidase, which acted on the substrate XGAL, and it broke it down, and the X material was released, and it just, that is blue, and that's how you understand. And see, that's my initials, see, that is why was my friend, my sister and me. See, that was our plate. See, it changed color. You can see it was blue. See, plasmids 3 and 4. They just mentioned plasmids here. The 3 and 4. Second transformation results. Those are the results of second transformation. 3 and 4, I guess. So that's that. Transformation via crossing method yet enabled the same beta-galactosidase. So if you bring the same genes together, with crossing, whether you're putting two plasmids together in the same cell, or you bringing two haploids, via crossing, so it depends on your lab's availability. If it is hard to work with plasmids, and it just whichever easier. See, and see, these are mated diploids. So this is say this one, let's say CNF1 haploid with another haploid carrying JAD CNF4. This combination worked. Same thing here. 1 hour after incubation. So that's the cellular machinery working, with substrate. That's the time, I guess, it took to generate color. Crossing of transformed strains resulted in beta-galactosidase phenotype. So it depends on your laboratory conditions. Whether because, like I said, it is hard to generate all these prototrophies and and all these selections, and ampicillin selection, and this selection, that selection. It's sometimes it's hard. So it depends on your the lab. You can use haploids, individually transformed haploids. So this data suggests both double transformation with plasmids and crossing, of different haploids enables co-expression of fused hybrid proteins, resulting in protein-protein interaction. Yet crossing method can be used. More effectively via replica plating, thus crossing multiple different strains in order to observe best interaction or prey. Provides time and advantage, like depending on your lab, so replica plating is like, you have a master plate, and with you use it. You use the master plate as a stamp, and then you use velvet fabric, and then you can make many copies of the master plate with the velvet. Use the master plate as an ink. Use the velvet block as a stamp, and then it just you can make many plates, 10 plates, 20 plates. Bam, bam, bam. So that should work better maybe. And I mean, depending on depending on your lab and depending on your protein. So it's a high throughput. So it's another way to do it. See, working with yeast is very, very advantageous. You can work with plasmids, you can work with haploids. That's why I'm excuse me if I just made mistakes or had a hard time explaining it, but just the idea is, if you're trying to study proteins, the yeast to hybrid system is a good way of doing it. You can have your own plasmid, and you can understand those protein interactions. And it says some of the limiting factors of this method is not every protein can be used in this analysis, such as proteins with high affinity to DNA. If the protein itself is directly binding to DNA, what's the use of it? Because it can to change the outcome. But it is a method that people use. And we already know that if the protein is located in the cytoplasm, then that it is not directly interfering with the DNA, because DNA, transcriptional proteins are different. They are often smaller, and they can go through the nuclear membrane. Because in eukaryotes, the nucleus is enveloped in a membrane too, the nucleus membrane. This is usually true for cytoplasmic proteins or membrane proteins, the ligand proteins and etc. Switching polarity of hybrids and group protein analysis may increase success rate of such interactions and complexes. So there's some things you can change. Polarity of hybrids and a good protein analysis. Like I said, it mostly works maybe for cytoplasmic proteins or membrane proteins. And that's it. That's the end of it. Thanks for listening. Please subscribe to my channel so that I can continue my work and explaining these methods. These yeast genetic methods are very useful for science. And listen, I mean, still we just see that we have many problems to overcome at this day and age. And these are the methods, the methods. Maybe they are useful, if I can explain, if you haven't heard of it. And please subscribe to my channel so I can continue explaining, teaching science. Thank you.